Next, we have Jeff Statland, who will be talking about getting ready for clinical trials.
All right, I want to thank the FSH Society for giving me the opportunity to speak to you today about clinical trial readiness. The last time I spoke at the FSH Connect meeting, which was just over 2 years ago, I spoke about the tools we use to measure the disease, outcome measures, and at the end of my talk I said, well, I think we might see some clinical trials in the next few years. And indeed, now it's 2 years later, and there's one clinical trial that's been active within the last year, and another clinical trial that's really set to begin. And you've just heard in this panel that there's a number of drug companies and really talented basic scientists who are looking at ways to actually alter the fundamental molecular defect that causes FSHD. And I think encouraging us at this time is some of the experience that we've seen in other inherited diseases of muscle and nerve, where Duchenne muscular dystrophy just got their first ever molecularly targeted therapy approved by the FDA for the disease, and spinal muscular atrophy has their first molecularly targeted therapy currently under review by the FDA. And the technology, the chemistry that they've used for both of those therapies, the antisense oligonucleotides, certainly might be applicable for treating FSHD. So I'm talking all about these clinical trials and what's going on. So why clinical trial readiness? Well, we as a community are stakeholders in the process of drug development. We need to facilitate therapeutic development, not just for one drug, but for all the drugs that may benefit people with FSHD. We need the tools in place so we can be confident we're not throwing out things which might actually work, or we're not approving drugs that aren't really going to help. Everybody who has a say in what goes on for treatments in FSHD needs to be part of this conversation. We're all at the table. We're all have an interest in that stake. This includes people in the pharmaceutical industry, people in academia like me, the basic scientists, your advocacy groups, and then you and all of your family members. So why plan now? Well, hand in hand with what the basic scientists are doing in the lab to develop new therapies, from a practical point of view, from a clinician who wants to run clinical trials, there's a lot of work that needs to be done. We need to ensure standard techniques for how we measure things, address gaps in our understanding of what happens clinically over time, develop the tools for the clinical trials, and make sure results are consistent when we implement them across multiple locations, and figure out what features predict how fast the disease is going to change. We need to advocate nationally but also internationally to get the resources to pursue research and pursue these cures. But most importantly, we need you. Without your help, without you volunteering for these studies, we can't make any progress at all. So it's really a process of innovation and collaboration. Early on, we need these people working independently. We want people thinking outside the box, thinking of new ways to measure the disease, new avenues for treatment. But at a certain point, ultimately, we need people to then come together and test these new ideas in large groups of people. We want things to work for everyone with FSHD. We want to ensure our assumptions about what our tools are measuring are actually true. If we can agree on common approaches, we can accelerate the drug development process. We can take the example of Duchenne muscular dystrophy and spinal muscular atrophy, where there was a collaboration between the advocacy groups, the clinicians doing the research, and the drug companies to really help fill in the gaps of what they didn't know and accelerate the overall process of drug development. We've made a lot of advances in our clinical trial tools, even over the last few years. But for each of the categories of tools I'm going to talk about, there's really still work that needs to be done. And when we think about the tools, they include things like biomarkers. These are things we measure in your blood, or we get from a tissue like muscle, or they can be other physiological measures. They can provide us proof of concept that a drug is reaching its target, For example, if it's a disease-directed therapy, maybe we're going to want to see those levels of DUX4 change. But if we can't measure that, maybe we want to see the downstream targets of DUX4 changing. And what you see in this picture is a picture of a muscle biopsy from a patient that's grown in culture, and DUX4 is that bright pink signal. Below that is some proof of concept that we can use the downstream markers of DUX4 to measure how things happen in a disease. This is from a mouse experiment where they were able to knock down the expression, to stop the cell from the mouse from producing DUX4, and you can see two of those downstream markers then also went down. We can use these biomarkers to monitor disease activity. It might be MRI, which can measure inflammation in the muscle, fat, or fibrosis. And what you see in this picture is just an example of a certain sequence on MRI where you can see that inflammation appears as a bright white signal. Or it can be something that we're measuring in your blood, a protein that might track with the progression of the disease. And this is an example of a study that was recently completed at Seattle and the University of Rochester. And what you see here are just four of the markers they selected, and you can see how they're relating to disease severity. It can be monitoring a physiological change. What you see pictured here is a device called electrical impedance myography. The FSH Society gave me some money and asked me if I would pilot this in people with FSHD. It's a really sort of simple device. The sensor, you can see it there, is small. It provides a sub-threshold current. You can't really feel it, but it tells us about the composition of the muscle. And on that bottom graph on the y-axis is the measurement of impedance, and on the x-axis is the amount of fatty replacement in the muscle. And you can see in the top left, there's an individual where on MRI the muscle looks very normal, and on the bottom right, there's an individual where the muscle's completely fatty replaced. So this may be a good indicator of what's going on inside the muscle. Biomarkers are important for these early studies as early signals before we can see changes in things like strength or function. But before we can use them, we need to standardize the technique, show we can get consistent results from multiple centers, and understand how they naturally change in people with FSHD over time. The other thing that we often measure are strength and functions, and intuitively these make perfect sense in a disease of muscle, right? We want to know if people's strength or function is changing. We have a number of ways for measuring muscle strength, and these include things like what we might do on you in clinic, where we just check how strong your muscle is at the bedside, but it can also include digital force transducers that give us a readout of how strong you are, or sort of space-age machines like you see there that give us a sense of force during movement. Functional tasks are very appealing when we think about changes that might be important to us because they're typical tasks that you do on a daily basis, like how fast you can go a certain distance, getting out of a chair, or walking up some stairs. There you see a pictogram of one of the common motor functional tasks that we do, where we have someone get up from a chair, walk a defined distance, turn around, come back and sit down, and you can see a boy there on a standard set of 4 stairs. You can combine these functional tasks into composite measures, and by doing that, you can get sort of an overall functional picture of how someone's doing at a given point in time. There's a number of standard instruments that have already been created to do this, but we've recently been working with help from the FSH Society to create one specific for people with FSHD, the FSHD Composite. And finally, you can even take some of the new modern technologies and instrument these functional tasks to get more information. Can you play the video on this slide for me? Can you activate the video? Right, and so what you see here is I've put on wireless motion monitors on this individual. He has them on his wrists, his ankles, and across his body. And you can see as he moves, what it's doing is it's transferring the information for how he's moving around his legs, the way his torso is moving in relationship to his legs, and the swing of his arm into actual numbers that we can analyze. And so you see in that graph there that there's a number of specific things it tells us, like how fast he's walking, the stride velocity, or how his arms are swinging. And the green color shows you that almost all of these numbers are different than when we compare them to healthy volunteers. But there's still a lot of work that needs to be done. We need to standardize our equipment and procedures, train our evaluators, and ensure we understand the relationship to the other features like genetics and age and gender. Finally, we have patient-reported outcomes, and we're asking the question of how a treatment impacts someone's life. These can be standardized instruments, and some of you may have taken some of the instruments like the short form 36 in the past. We've also been developing one specifically for FSHD. I did this with in collaboration with my colleague Chad Heatwall. This is the FSHD Health Inventory. He started with individual interviews with people who have FSHD. He then took the most common things he heard and created a survey that he sent out to over 300 people. Many of you in this room may have completed this survey where they ranked whether or not they had a symptom and how impactful it was to their life. And that's how we developed the categories for this questionnaire. But we still have work to do here to establish the common instruments which cover a full range of disability, understand the normal variability from year to year, and determine if there are differences from a region to another region in the country or from one culture to another. So one of the things that's of vital importance for us is to understand the natural progression of the disease, the ability to understand the normal progression in FSHP and how it impacts the type of tools that we're using can help prevent us from rejecting a drug or a therapy when it may actually work. This is an example from Duchenne muscular dystrophy from one of their earliest molecular-based therapy clinical trials. They had chosen how far the boys could walk in 6 minutes. Each of those lines is a boy. The direction is how they changed over 1 year. And I think what you can see are some are improving, some are stable, but about 300 meters, suddenly they stopped walking. This was unexpected, and it created a high amount of variability in this clinical trial and obscured their ability to see a treatment effect. So understanding the natural history is going to help us design more efficient clinical trials. It will reduce the burden on people like you who are participating by allowing us to reduce the number of people required if we have a more consistent sense of how the disease is changing, and help us design the confirmatory studies. There's still a number of gaps in our understanding which need to be addressed. How does genetics, your baseline functional status, and demographics influence how you change over a year? And are there simple tools we can use to predict how someone might do over the next year? Finally, a really important question that we need to know the answer to is how big a change is going to be meaningful. Ultimately, understanding what kinds of change in these tools that we're using are going to be meaningful will help us make the argument with the approval agencies like the FDA to allow them to market a drug. Short of hard outcomes like living and dying or the time required to have to use a wheelchair or non-invasive ventilation, we need other ways to determine how big a change is going to be meaningful. And this will depend on what the treatment is expected to do. For example, a drug designed to help with pain doesn't really need to improve how you're walking. We can try and understand this by using statistical techniques, using questionnaires, or relating a change in one measurement to a change in another. And again, in order for us to do this, your voice is going to be very important. So finally, what we've done recently, seeing all of the clinical trials that are starting up or coming soon, we've collaborated. There's 7 large academic centers across the United States. The FSH Society has helped us do this in private funding to form an FSHD clinical trial research network. The overall goal of this network is to hasten drug therapeutic development for people with FSHD. We plan to work with all of the stakeholders, the drug companies, researchers, advocacy groups, and you, to address gaps in our understanding of the natural history of FSHD and develop the tools we're going to need for clinical trials. The other thing that we're going to do is make these large clinical data sets available to anyone with an interest for developing therapies in FSHD. This network is getting started now, and your participation can actually make a big difference. You can either participate directly in the studies, or there's other ways you can participate. You can give money to the FSH Society, or you certainly can contact me, because we're looking to have people involved in the activities of this network. I want to thank you very much for giving me the time to speak to you.
Thank you so much. Maybe we have time for one question for Dr. Statland, if anybody. Okay.
So I would love to do every single clinical trial that comes out. However, it's expensive to fly to these places. I tried doing the ATIRE, but then it was like, oh, you have to fly twice a week to Baltimore, and there's no travel funding. So do you have an answer for that?
Do I have an answer for that? You know, so part of what you're asking is in an actual clinical trial, is there money to have people travel? And this will depend on the company running the study. We do a lot of studies at our center. Some of the studies have money, so we can help people travel to the site if they need to. Others don't. It sometimes depends on the size of the drug company, how much money they have, and how big a study they're planning. As these studies start to get closer to trying to seek approval, they tend to get larger. What you'll find is going to happen then is there'll be more sites across the country, so you're more likely to have one close to you, and there'll probably be more funds to help with travel.